Hi all, I'm back. I'm just going to do a short lecture. Again, we'll only get through just a portion, probably about 30 slides of the peripheral nervous system. I know that this is going to go a little over our normal lecture time, but I want to keep us current with where we should be. So what I'm going to do is I'm going to go ahead and just cover the very first half of the peripheral nervous system. It's, again, the more basic portion. And then when we get into the cranial nerves, I'll actually do that when we come back from the Thanksgiving break. So again, I'm just going to pick my pen color. We'll do red today and make sure that I have my pen chosen so that way then I can write on the slides. It helps me to annotate through the lecture. So again, this is going to be the peripheral nervous system. We've done the central nervous system. We spent a long time on the central nervous system. We did both the brain and then a brief lecture on the spinal cord. And now we're going to move into the peripheral nervous system. And the peripheral nervous system is everything else. So everything but the brain and the spinal cord. So this is going to include your sensory receptors, your peripheral nerves, including your sensory neurons and your motor neurons, any ganglia that are associated with that, and the motor endings. So basically the effectors where your motor neuron ends, and then now is going to either release neurotransmitter or directly stimulate your receptors. Again, when we talk about your motor endings, your motor neurons are going to be stimulating either a skeletal muscle or a gland. So again, those are the big things that your motor neurons are going to do: stimulate and excite either a skeletal muscle or a gland. And this helps to provide, again, a link to and from the external environment to, again, the brain. So this helps to provide that nice link, is the peripheral nervous system. So just as a review, we start off again, here's the central nervous system with the brain and the spinal cord, and the peripheral nervous system again is going to send information to and carry information back from the central nervous system. So when we take the peripheral nervous system, we can branch it then into the sensory division, or the afferent, right, division, which is going to be sensory information, afferent neurons bringing things back towards the central nervous system. And then we can branch the other half into the motor division, which is the efferent division, neurons that are exiting. Remember, exiting the peripheral nervous system. So the peripheral nervous system is the one that we branch into everything, because remember, the central nervous system is just the brain and the spinal cord. Wow. I have not gotten any better at writing with this. So this is brain and the spinal cord, okay? And then the peripheral nervous system is the one that gets all crazy. So the peripheral nervous system is the one that branches into the sensory or afferent. So remember, afferent is those neurons carrying information to the central nervous system, and motor division consists of your efferent neurons, your motor neurons, which are exiting the central nervous system and carrying information away from the central nervous system. So then the motor division can be branched even further, and the motor division can be branched into the autonomic nervous system here, and then the somatic nervous system. The somatic nervous system, again, is going to be the body nervous system. So this is going to be, again, where you're going to be, this is going to be where you're stimulating. Oops. And this is my symbol for stimulating, although it's coming out pretty sad looking there, but an S with a T above it. So that symbol means stimulate. So our somatic nervous system is going to stimulate our glands and our skeletal muscles. And then so that's what your somatic nervous system is going to branch. Those motor neurons are going to glands and skeletal muscles. Your autonomic nervous system, which is the other branch here, is going to stimulate your vital organs. So again, like increasing your heart rate when the sympathetic division is stimulated, or in the parasympathetic, decreasing heart rate and stimulating gastric motility. So autonomic nervous system, again, is really affecting more of your viscera. So the autonomic nervous system then is what we're going to get into when we come back from the break, from the Thanksgiving break, is going to branch into the sympathetic and the parasympathetic. The sympathetic, remember, being your fight or flight, and your parasympathetic being your rest and digest. But again, we'll get more into that when we come back from the break. So we're going to focus again on mostly the somatic nervous system here. Well, we'll focus on both the sensory and motor, but when we get into the motor division, we're going to focus on the somatic nervous system. So let's talk about sensory first. So when we talk about the sensory nervous system, there are all different types of sensory receptors, which are just structures that are specialized to respond to stimuli, whether the stimuli be internal or external. So here's some of your, examples of your sensory receptors. These are your special senses. So you've got, again, your rods and cones that make up your vision receptors. We'll talk about some of your hearing receptors, like the tympanic membrane and things like that are it's going to be mostly your vestibulocochlear apparatus. So inside your vestibulocochlear apparatus, you have the actual sensory receptors that will be kind of transmitting the information for hearing. We'll get into that. We'll get into that in our special senses. We've got smell and taste and touch. And we'll talk about the different touch receptors as well today. So activation of your sensory receptors is going to result in depolarization in those sensory receptors, and that can trigger impulses in the sensory neurons that are sent back to the central nervous system. So that's, again, the goal is to activate a sensory receptor that will trigger an impulse ultimately in the sensory neuron that's going to go back to the central nervous system. And the realization of these, or I should say the perception of the stimulus, is going to take place in the brain. So the brain, again, is going to take in that information and will formulate the perception of our different senses. So again, the perception of smell, the perception of hearing, things like that. So we can classify receptors in different ways. We can classify, one of the ways that we can classify them is we can classify them by stimulus type. And this is pretty easy, so this will be a nice, easy question for you on the exam. So mechanoreceptors are just like they sound. They're going to respond to some type of mechanical stimulus, whether it be physical touch pushing down on the receptor that causes the gate to open, or pressure, vibration can cause some of those mechanoreceptors to the gates to open. And we're talking about these mechanical gates that would allow ions in, right? Then some of them again respond to stretch, some of them respond to itch. So these are all mechanoreceptors respond to mechanical stimulus. Thermoreceptors, just like it sounds, thermoreceptors respond to changes in temperature. Photoreceptors are going to respond to changes in light energy. So again, we see photoreceptors inside the retina, especially the rods and the cones. Chemoreceptors, just like it sounds, responds to chemicals. So again, we have chemoreceptors that are going to pick up the chemicals for odor, for smell. So they'll pick up chemicals changes in the air, pick up chemical changes in the mouth. That, again, is going to help formulate our taste. Chemical changes inside the blood. So those are chemoreceptors. Nociceptors, remember, nociceptors are our pain receptors. So these are sensitive to pain-causing stimuli. So we can classify our receptors again by their stimulus type, or we can also classify our receptors by location. And the two classifications for location are exteroreceptors and interoreceptors. And again, this will be a nice easy question for you. So exteroreceptors are simply any type of receptor that's going to respond to stimuli that's arising outside the body, hence the extero. Okay, so you can think extero, external. They're responding to stimuli that are external, outside of the body. So these are going to be things that are found near the surface. Again, sensitive to outside touch, outside pressure, pain from an outside stimulus, temperature changes at the surface, okay, of the skin. So those are, and this is going to include our special senses. So again, our special senses, so our rods and cones are responding to light coming in from, again, outside the body. Sound waves, okay? Our hearing is going to respond to sound waves coming in from outside the body. So these are all examples of exteroreceptors. Interoreceptors are just like they sound. They're going to respond to stimuli arising internally, internally. So they're going to respond to stimuli arising within the body. These are going to be found, these types of receptors will be found in our internal viscera, so mostly around our organs and blood vessels. So they're going to be sensitive to chemical changes, again, within the blood, or chemical changes within organs, stretch within organs. The bladder gets stretched, right? So there are receptors, interoreceptors, that detect that, and temperature changes inside the body as well. So interoreceptors, again, are all inside the body in our viscera and our blood vessels. And then we also have our proprioceptors, and these are going to respond to stretch of organs, basically the degree of stretch of an organ that the proprioceptors occupy. So these are going to be found in skeletal muscles, tendons, joints, ligaments, connective tissue that's going to cover bones and muscles. And the reason that they're different than your interoreceptors is that these are proprioceptors. So they're constantly advising the brain of one's movement, so whether it be in a joint or a tendon. And this helps with our proprioception, our ability to perceive where our body parts, especially our limbs, are in relation to the rest of our body as they're moving. So these proprioceptors are kind of separated out as separate receptors by location, because they're in these special locations, again, tendons, joints, ligaments, skeletal muscles. We can also classify receptors by structural complexity. And this is pretty easy because the receptors are either classified as simple or complex. Most receptors are simple, and most of the receptors that are simple will include either an encapsulated or an uncapsulated variety, and I'll talk about these in just a minute. So and I'll talk about all of our different types of simple receptors. Complex receptors are going to be our special senses. So this is going to be our very last lecture. Okay. So we're going to talk mostly about our simple receptors here, but I want you to recognize that your complex receptors are your special senses. So this is our very last lecture on special senses. So what are our special senses? Our special senses are vision, hearing, equilibrium, smell, taste. So we'll go over those again at the very end of the semester. So let's talk about then our simple receptors. So with our simple receptors, remember, we have unencapsulated ones and then we have encapsulated ones. Our unencapsulated ones are pretty easy. We have free nerve endings of our sensory neurons. So sometimes, again, we have these sensory neurons and we just have these free nerve endings. A lot of them, again, can be associated with touch. We can have some of these located in our tendons, some of them, again, in our dermis for touch, some of them in joint capsules, things like that. And there are different types of free nerve endings. So we've got thermoreceptors, chemoreceptors, so all of those. So again, there's quite a few different types of free nerve endings. And again, they're a non-encapsulated, simple. I just want you to recognize that free nerve endings are a non-encapsulated, simple receptor. Our tactile Merkel cells or Merkel discs, I don't know if you remember those from when we did skin. So again, those are going to be in the basal layer of the epidermis, and they're going to detect light, very light pressure touch. So those are, again, a simple non-encapsulated receptor. And then our hair follicle receptors, we have, again, receptors that go around the hair follicle that will detect hair deflection, movement of the hairs. And those are also free nerve endings, or I shouldn't say free nerve endings, but they're free simple receptors. So they just have these nerve endings that go around the hair follicle. So those are the three types of non-encapsulated simple receptors. The rest of the simple receptors are actually encapsulated ones, which makes sense because we know about most of them. So if you think about it, Meissner's corpuscle we know is in a capsule. Our Meissner's corpuscle again is, we found those at the dermal papilla, if you remember. Those are going to detect light pressure, again, light pressure, light touch. We have the Pacinian corpuscles again, which is in another encapsulated corpuscle here, and that's going to detect deep pressure, right? That's going to detect deep pressure and vibration. You have your Ruffini bulbs or your Ruffini endings, and these are going to be deep pressure receptors and stretch receptors that we're going to find mostly in our joint capsules. You'll find some deep in the dermis and hypodermis, but we'll find a lot of these in our joint capsules. Then we have muscle spindles. Muscle spindles, we have already kind of talked about these when we did reflexes. And these are going to be receptors that wrap around the muscle fibers and detect stretch. And so they're going to be encapsulated. And then our tendon organs will also, you can see, here's the capsule for that. These will be encapsulated, and these are going to be detecting our proprioception. So especially on our tendons, they're going to be detecting movement of the tendon, stretch of the tendon. And then joint kinesthetic receptors are going to be in our joints. Again, we haven't really talked about those, so I'm less concerned that you know about those. There's not quite as many of them. But I want you to be able to recognize that the rest of these are your encapsulated simple receptors. These are all encapsulated simple receptors. So from sensation to perception. So remember, our survival will not only depend on our ability to sense, right, our sensory receptors stimulating our sensory neurons, but then perception, how we make sense of all that sensory information in the brain. So sensation literally is the awareness of changes in our internal and external environment, but perception is our kind of interpretation, our conscious interpretation of the stimuli. And these are great images to kind of show you the difference again between sensation and perception. So sensation, again, being in these images, are that the image, light and dark, the information that's forming this image, again, the light and darkness, is being sent into the eyes, into the retina, and being detected by our rods and cones in forming that image. However, as that information is sent to the brain, it's the brain that makes sense of it, right? In that visual association area. And so that's our perception of what we're seeing, what's coming into the brain through the optic nerve. And so again, that's the idea of when you're looking at this, do you see this and does your brain process it and perceive it as a vase, or does it perceive it as, two individuals facing each other? So again, the same thing as you look at this elephant, does it look normal to you, or does it look abnormal in terms of where the feet are? So again, just kind of some fun things in terms of sensation and perception. We'll get more into that in the special senses. That there's a lab on that, and we'll have some fun with some of the ways in which you can trick the brain in terms of your sensory and processing perception. So input then comes into the brain from these exteroreceptors, proprioreceptors, and interoreceptors. And the kind of three main levels of neural integration in the somatosensory system, again, because we're talking primarily about sensory information being brought in here. There's the receptor level, the sensory receptors that are actually, again, initially detecting the changes and being stimulated. Then there's the circuit level where those sensory neurons have to move up to the brain, so those ascending pathways. And then there's the perceptual level. The perceptual level is the neuron circuits that are in the cerebral cortex that are involved with, again, the actual perception of those different stimuli. So that's all this is showing is, again, we've got the receptor level where we've got our, initial sensory reception being sent back then through the sensory neurons and then up the ascending pathways. Here's our circuit level, right, as we're going up the spinal cord and then into higher areas of the brain. And then as we make it into the higher areas of the brain, this is our perceptual level once we get into the somatosensory cortex. So, and this is just kind of talking about basically these different receptors, how these different receptors work. So remember that these receptors will be stimulated and they're going to be generating these graded potentials, and how much stimulus how much of a stimulus is received will determine how much of a graded potential will occur and whether or not an action potential will be initiated in the sensory neuron. So again, that will determine whether or not information is sent back to the brain. So again, each of these sensory receptors has kind of can be stimulated at different levels, and the more they're stimulated will determine whether or not we actually reach threshold in that sensory neuron. So, and it's kind of interesting that we haven't talked about this. The Marieb doesn't go quite as much into this, but some of the sensory receptors are actually separate cells, and they'll release neurotransmitter to the sensory neuron. And then some of the sensory receptors are actually part of the neuron. So the sensory receptor itself will receive the stimulus, and then a graded potential will occur. And if there's enough of a graded potential that occurs to bring the resting membrane potential again to threshold, then an action potential will be generated in the axon. So again, kind of interesting just to see how sensory neuron would actually be brought to threshold. This is kind of fun because when we talk about sensory receptors can adapt. So adaptation is receptor membrane's ability to become less responsive. And the best way I can describe adaptation is, let's say you walk into a room and you either hear, like this time of year, if they're running the heat, you can hear the fan as you walk in the room, and it might sound kind of loud. You might think, gosh, that's a really loud fan for the heat, or the fan from the projector. And then as kind of the lecture goes on, you don't notice the sound by the end of it. And so what's happening is that's your adaptation of sensory receptors. So literally your receptors are becoming less responsive. Another example of this would be you walk into a room. So this always happens when I teach marine biology and we're dissecting, but it'll happen for you guys next week when we're dissecting the brains. You'll walk into the room and you'll be like, oh, it really stinks in here. But by the end of the lab, you don't even notice the smell at all. And then you might go home and smell your clothes and then notice the smell in your clothes again. So again, that's the idea of adaptation of those sensory receptors. Your sensory receptors are becoming less responsive. And the receptor potentials can either decline in frequency or even stop altogether sometimes. So, and that'll happen like when you put clothes on. You might first notice the feel of your clothes, and then, maybe a minute or two later, you don't even notice that you have clothes on. You can't, you're not like constantly feeling them rubbing up against your skin. So again, that's the same idea of that adaptation of sensory receptors. So receptors responding to pressure, touch, smell are all going to adapt very rapidly, very quickly. And that's why, again, you'll walk into a room, notice an odor, and then very quickly you won't notice it after a while. Receptors that respond slowly include things like your Merkel discs, deeper in your skin, Ruffini corpuscles, which are detecting, again, pressure and stretch, especially in your joints. So again, if you have joint pain, that's part of the reason why you're going to constantly detect that pressure with those Ruffini corpuscles. A lot of your interoreceptors that respond to chemical levels in the blood won't adapt very quickly. We don't want them to, because we want them to help us to maintain homeostasis. So we want them to continually send information back to the brain saying, whoa, chemicals are, out of whack in the blood. Pain receptors and proprioceptors don't exhibit any real adaptation, and this is why pain is such a pain, right? Is because we don't get that adaptation of pain. So the pain receptors don't adapt. Now you may say, but I've had, chronic pain before, and it ends up being a little bit better over time, and that's actually how your brain is processing that. So what happens is you're still getting the pain receptors are still constantly being stimulated, and they're sending information to the brain and saying pain, 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 pain. You have pain in your hand, you have pain in your hand, but your brain just stops listening because it gets that constant reception. So the adaptation for pain actually happens in the brain and not with these pain receptors. But pain receptors don't actually exhibit adaptation. So they'll just continually be stimulated if you have pain in that area. So these are the two, again, types of receptors. So tonic receptors, again, are just basically what we talked about. Tonic receptors are going to be any type of sensory receptor that's going to adapt slowly or doesn't adapt. So again, these are things that we talked about, like the Ruffini's corpuscles or the pain receptors. Those are going to be things that either don't adapt or are gonna adapt really slowly, like your Ruffini corpuscles will adapt really slowly. Versus your phasic receptors, so they're tonic because they give you that more tonic graph here. Phasic receptors are going to produce signals when the stimulation changes. So they're primarily alerting you to changes. So these are going to adapt very rapidly. So things like your poscinium corpuscles, your odor receptors, things like that, are again going to allow you to adapt very quickly to those changes, or to those well, I shouldn't say changes, but if you're getting a stimulation there, if the stimulation's constant, they'll adapt very quickly. And again, some of them are phasic. They'll only alert you to a change. So then we kind of already talked about processing at the circuit level, so I'm not going to go back over because we've already gone over first order, second order, and third order neurons in the last lecture. So again, you can review that, but I won't make you go through that again. We've gone through the first order, second order, and third order. Again, first order bringing information into the spinal cord or the brainstem medulla oblongata, especially second order bringing it from there on up to the thalamus or the cerebellum, where it ends at the cerebellum. And then the third order, again, is going to take it from the thalamus to the somatosensory cortex. Okay, and then the thalamus, again, we talked about it, projects mainly to the somatosensory cortex, sometimes again to sensory association areas as well. And that is where we're going to, again, get our perception of the sensation. So again, it's going to be in the cortex that we're going to get our perception, especially in the somatosensory cortex and then in the sensory association areas. So this gets into the kind of main aspects of sensory perception. We're not going to get into all these. Are interesting. You can read over them. The perceptual detection, detecting that a stimulus has occurred, the magnitude, how much of a stimulus is acting up, spatial discrimination, identifying the site or pattern of a stimulus. So these are kind of interesting, but these are just mainly talking about kind of some of the aspects of sensory perception. So you won't have to know them for the exam, but again, they're kind of interesting if you want to look over them in terms of, again, our sensory perception that occurs there. Same thing here. Sorry that I meant to cut these slides. We already went over the endoneurium, perineurium, epineurium, so I won't make you go through that again. And we've already kind of talked about classifications of the nerves, of the sensory and motor divisions. Again, sensory being that you have afferent, sensory neurons are afferent. That's just reminding you of that. They're carrying impulses to the central nervous system. Motor nerves are efferent, exiting the central nervous system, carrying impulses away. And mixed nerves are the majority of our nerves. So most of our nerves are a mix of sensory and motor fibers. So they're carrying impulses to and from the central nervous system all in one nerve. Because remember, a nerve is just a big bundle of axons, right? Of neurons. Ganglia, remember ganglia are these little outcroppings of neuron cell bodies that we see in the peripheral nervous system. Ganglia associated with our afferent nerve fibers, our sensory ones, are going to be the dorsal root ganglia. So remember, again, we have those unipolar neurons. I keep reminding you of this, right? These unipolar neurons with those weird, remember the unipolar neurons look like this with these weird cell bodies hanging out here. And so as those sensory neurons come into the spinal cord towards the central nervous system and towards the spinal cord, as they enter in, they're going to branch, and the sensory neurons are going to enter the dorsal root or the posterior root, and there's going to be a root ganglion because we have these lovely cell bodies. So that's going to be where we're going to have our dorsal root ganglia or posterior root ganglia, whichever one you, again, like to go by. And then we are going to talk about, and we're going to get to it in the autonomic nervous system. There are ganglia associated with efferent or motor nerve fibers, but these are for our autonomic nervous system, and that's why we haven't talked about them yet. So when we get to the autonomic nervous system, we'll talk about our autonomic ganglia. So we'll get there. And this is just a kind of, again, another reminder. Remember in the peripheral nervous system that we can have some regeneration of nerve fibers. If the damage to the nerve tissue is severe because the mature is really serious and severe, again, then we're out of luck. Because remember that these mature neurons are what we call amitotic, and that means they cannot undergo mitosis. But if the body, the soma, the body of a damaged nerve is okay, it remains intact, then damage can be repaired in the axon, in the peripheral nervous system. And so remember, this happens because we can form those regeneration tubes from the neurolemma. So this is the easy part. For regeneration of nerve fibers, you guys only have to remember 3 things. 1, macrophages are going to come in and clear up the old debris. So that, if the nerve was damaged, the axon's damaged, they're going to come in and remove the old debris, kind of clean it out. Then the Schwann cells, okay, the neurolemma, remember that neurolemma? That neurolemma is going to form the regeneration tube. And then those Schwann cells will then secrete growth factors, and that will stimulate the axon to regenerate the damaged part. So that's it. That's it. 3 steps of the regeneration of nerve fiber. Very easy in the peripheral nervous system. But again, remember, this can't happen in the central nervous system. And why can it not happen in the central nervous system? Because, remember, we have oligodendrocytes that are myelinating our axons. And those oligodendrocytes do not have a neurolemma, so they can't form the regeneration tube. So this will only happen in the peripheral nervous system. And this just, again, goes over a little more detail. Again, I only care that you know these 3 major steps here. And so again, this kind of shows the macrophages coming in and cleaning it up. And then again, the neurolemma forming that regeneration tube. And as that neurolemma forms this beautiful regeneration tube here, then the axon can start to repair itself, especially as that Schwann cell secretes some growth factors. So I'm going to end here just talking about cranial nerves. And so this would be kind of the very tail end of our peripheral nervous system lecture. So we've talked a lot about sensory neurons, and mainly we've talked a lot about nerves that would have gone back and forth to and from the spinal cord. But we also have what are called cranial nerves. And these are 12 pairs of peripheral nerves that are going to arise from the brain. So they'll attach to the brain, either the forebrain or the brain stem. And these cranial nerves will have sensory, motor, or they'll be both sensory and motor in a package. Most of them sensory and motor in one package of a cranial nerve. Only two of them, and we'll talk about those, are purely sensory. So what I'm going to do is I'm just going to introduce how to identify the cranial nerves, and then we'll get into it in lecture next week. So each cranial nerve is numbered 1 through 12, and you number them with the, again, Roman numeral form of numbering. So, and they're named from rostral to caudal. So again, if you take the brain, they're named kind of from front to back, and I'll kind of go through this on the brain in just a minute. So there's a couple different tricks that you can use to remember the cranial nerves, the first letter being the first letter of each cranial nerve. So, on occasion, our trusty truck acts funny, very good vehicle anyhow. That was one that the book had. Oh, once one takes the anatomy final, very good vacations are heavenly. There's another one. The one that I usually teach is the to touch and feel, very good velvet, aha. This is the clean version. You can always come up with your dirty versions. I'm sure you can be creative. But this seems to be the one that many of our anatomy students like. And so this will be the one that I use. Again, you can alter this however you want, whatever way you can find that gets you to remember the cranial nerves. So I'm going to show you how this works as to touch and feel, very good velvet, ah. So the first O of O in is going to be your olfactory nerve. So here's going to be your olfactory nerve. Okay, so there's the first O. The second O, remember. So we've got 3 of them. So the second O is going to be the optic nerve. And so here again is going to be your optic nerve here. And then there's the third O. Third O is your oculomotor right here. Okay, so there's to, the first T of that is going to be to, trochlear nerve. So here's the trochlear nerve. Okay, and then there's touch, which is the trigeminal. So here's your trigeminal nerve. And, okay, it's going to be the abducens nerves right here. Okay, then there's feel is going to be the facial nerve right here. Very is going to be the vestibulocochlear nerve right here. Good is going to be the glossopharyngeal nerve here. Velvet is going to be the vagus nerve here. And aha is accessory nerve, is the A for ah. And then ha is going to be the hypoglossal nerve right here. So there are your basic cranial nerves. What I'm going to do is I'm going to end here, and then I'll go ahead and recap the cranial nerves for you when we come back from the break.